All right, today we're finally putting everything together. So, transcription, just a quick review. In transcription, we went from DNA that was in the nucleus and it turned into mRNA, which has the ability to leave the nucleus because it's small enough and it's only one strand. So, it can leave and go out into the cytoplasm. So, that is where transcription ended. And today, translation is going to pick up where this leaves off. So right now, our mRNA and its adventure is just out in the cytoplasm. And we're going to find out how today that little mRNA is going to turn into a protein. So before we can do that, let's look at some terms. Protein synthesis is simply the making of proteins through this process of both translation, the one we're looking at today, and the one from Friday, which was transcription. Now, for some of you, Friday was not when you looked at transcription, it was now just today. So in order to make a protein, we have to go through both this process and this process. Proteins in and of themselves, and this is going back to macromolecules, cats like plenty of naps, carbs, lipids, proteins, and nucleic acids. So we're looking at nucleic acids, but what we're now learning is that nucleic acids code for making proteins right here, because proteins are simply a bunch of amino acids linked together by something called polypeptides, which is just the bond that's between them. What you need to know, little review, is that proteins are made up of amino acids, and there are 20 different ones in the body that can make proteins. So when you're buying protein powder, you're usually buying amino acids that are hard to find in foods that you eat regularly.
You can also buy amino acids.
That's what you said.
You said protein.
Right, but you buying the protein powder. Sorry, that's Mr. Sandberg. We have to get more protein powder too, by the way. We buy protein powder, but it contains amino acids in it.
But you can buy
the amino separate.
Supplement.
Oh, I see what you're saying. Yes, and the amino acid supplement. Yes, yeah.
Good talk.
So you do get a lot of them from your food, but the reason why people buy them, and yes, we buy them, is because they contain different ones that is just difficult to find in your food. So the order of the amino acids shown down here, here's some examples of amino acids, is going to determine the type of protein and what it's going to do. Is it going to be an enzyme? Is it going to go and be in your mouth and help with digesting your food? That's all based on the amino acids that get linked together. So fast forward here back to our genetic code. mRNA, keep in mind we have uracil and stealth-thymine, is just like DNA in that it can be read, and it's a code. And this code is read very specifically in letters of 3. And these letters of 3 are called a codon. And each codon will make one amino acid. This is going to seem kind of abstract today. When we go through it together as a whole class, it will make way more sense. But if this was our mRNA, keep in mind it came from DNA. This originally was pulled from DNA. If I wanted to read it then in these codons, which are just three nucleotides, because this would be attached to sugar, attached to phosphate, just like DNA. If I'm going to read this in groups of 3, it would be broken down like this, and that's what we have done here. So it's broken down into groups of 3, and then you read these codons, and you use a chart to figure out what amino acid they code for. So if I have 3 codons, 1, 2, 3, then I'm going to have 3 amino acids, 1, 2, and 3. So you're not going to have to memorize the chart. You'll be given the chart and provided with everything you need to know to be able to be successful. So now let's kind of cruise forward, and here's just some tips when you are doing this process of translation. Because we have 20 amino acids and 3 different codons that they're in and 4 different bases, there are 64 different codon combinations, and these are universal for almost all organisms. So for us, for cats, for fish, for deer, we all have the same amino acids. It's just how they're linked together and form different proteins that make it different for them and us. Some codons will code for the same amino acid, and you'll see that in the chart. And And then there's a codon that is going to be something called our start codon. This is going to let the body know to start making a protein. And then there are 3 codons down here that will code for stop. These are going to be built into your chart, so you're not going to have to memorize those. And if, to double check my chart, they're not on there, we just write them in. So you know which one is the start and which 3 are the stop. Because here they all are, the 3 different stops. So as soon as you get a stop, sorry about that. As soon as you get a stop, you don't have to continue making your protein. You are done. So translation. So what happens in this process? So in translation, we're taking mRNA, those from the DNA, and we are turning it into, again, a protein. So we pick up where we left off. mRNA leaves the nucleus and it goes down here into the cytoplasm. And now you'll see we're introducing this guy, which is a tRNA. The tRNA has an amino acid on it, and it has something called an anticodon, which we don't spend a lot of time on. It's going to be hard for you guys to read that. We don't spend a lot of time on that one because that one's kind of a little bit higher concept. But we have an anticodon on one side, amino acid on the other. So what's going to happen is that anticodon docks next to, I knew that was going to happen. I can't mark down there. Next to the codon that's on mRNA. So the codon has to match the anticodon. That's why there's an anticodon there. And then that amino acid is going to dock. And there it is right there. And then the next tRNA would come in and dock on the next the anticodon on the tRNA would come in and dock on the next sequence of codons and drop off its amino acid, and the process would just continue, which you can see here. So we end up with a bunch of these amino acids linked together, which the end result here, there it is, a chain of amino acids, is a protein. So your body continues to do this process at a ribosome. I don't have that on here anymore. Out in the cytoplasm at a ribosome is where all this would be taking place. And it continues to do this process of translation until it hits that stop codon, of which there are 3. And it can only start when it hits the start codon, which is A, U, and G. I promise once we do practice with this, it will make a lot more sense, and we'll be practicing this tomorrow. The video that will bring this to life is after this, just like we've had in the same sequence, so that you can understand this a little bit better.